Last chapter, we discussed the three major cell types briefly. This chapter is going to go into further detail about bacteria and archaea, your prokaryotes. Let's first discuss how these guys differ from the eukaryotes. They have DNA, but they store it a little differently because they don't have a nucleus, nor do they have histone proteins. Their cell wall differs as well. These prokaryotes may have peptidoglycan, as what we see in bacteria, or they may have some other type of carbohydrate. These guys don't have organelles, but that doesn't mean they're limited. You'll be surprised to see how they can do so much with so little. We will start off by discussing structures seen in bacteria. Not all bacteria are the same, but they'll all have things like a cell membrane, cytoplasm, ribosomes for protein synthesis, a cytoskeleton for support, and chromosomes. Not all, but most bacteria contain a cell wall, along with the glycocalyx, which is a carbohydrate-rich substance. Some additional structures that we will also discuss are not found in every bacterium. We will discuss each of these in just a bit. This is a nice picture that depicts structures seen in bacteria. Keep in mind that not every type of bacterium has all of these structures. Here we can see those that all bacteria have, and as well as the additional structures. Typically, bacteria tend to want to act alone. A single bacterium has the ability to survive by itself. However, occasionally you'll see them come together as a community and form biofilms, which are problematic. They're difficult infections to treat with antibiotics. Of course, we've discussed their size, and they're relatively small. A cocci is a spherical-shaped bacterium, whereas a rod is pill-shaped. What type of nutrients they acquire, as well as what types of genes they possess, can also determine the size and shape of the bacterium. These differences lead to pleomorphism. This table outlines a typical bacterial shape seen. Your spherical cells are called cocci, and organisms like Staphylococcus aureus take on this shape. We have rods, also called bacillus cells. One familiar bacillus cell to you would be E. coli. Vibrio cells are rods that take on a curved appearance. Vibrio cholera, the bacterium that causes cholera, is this shape. There are spirillum, which are helical or curved. An example of this would be Campylobacter jejuni, which causes gastrointestinal issues. The spirochetes are spring-like, and Treponema pallidum, which causes syphilis, is an example. Last but not least, branching filaments, which resemble yeast. In addition to the shapes, there are also different arrangements that bacteria can take on. They can be single, diplo, which means in pairs. They can be in groups of 4 or clustered like grapes, which would be a staff arrangement. They can be in a strepto arrangement, which resembles pearls on a string, or like a cube. The arrangement of the cocci-shaped bacteria come from what types of planes they divide in. If they divide in one plane, we will see a diplo or a strepto arrangement. With division in 2 planes, we see a tetrad or a Sarsana arrangement. And we get a staff arrangement with divisions across multiple planes. Bacilli can be arranged similarly to cocci, but instead of a staff arrangement, they have a palisade arrangement. This is an irregular grouping of them. We typically find spirilla in chains, and spirochetes generally are found as individual cells. Let's start discussing the various structures that we can see with bacterial cells. We will first start with the external ones. Some bacteria have the ability to move and do so using flagella. Some are also fortunate enough to utilize structures like fimbriae and pili to attach to surfaces, which helps them to cause disease. They may also have nanotubes and nanowires to communicate. There are three major components of the flagellum, including the filament, hook, and basal body. In a gram-negative bacterium, the basal body spans across the cell membrane, cell wall, and the outer membrane. The hook, along with the basal body, helps to support the filament, which is used for motility. Bacteria can have various types of arrangement for flagella. They may have just a single one in a monotrichous arrangement. They may have many that resemble a horse's tail in a lophotrichous arrangement. And sometimes they may have them at both sides of the bacterium. In a peritrichous arrangement, a bacterium may have flagella all over its surface. Here in A, we can see a monotrichous arrangement. B shows us a peritrichous arrangement. C indicates a lophotrichous arrangement. And D represents an amphitrichous arrangement. So how do these bacteria use flagella to move? They do so using a process called chemotaxis. In positive chemotaxis, a bacterium will move towards a source, usually a nutrient or food source. On the other hand, a bacterium could move away from a source, such as an antibiotic, during negative chemotaxis. They do this using runs, in which their flagella rotate like hands on a clock, counterclockwise, and tumbles, where they reverse the motion of the flagellum and basically do backflips. A shows us motility with a single flagellum, while B shows flagella in the peritrichous arrangement. Let's look at this process of chemotaxis. The pink represents tumbles, while the green represent runs. In A, there is nothing positive or negative to catch the eye of the bacterium, so it just kind of runs and rolls around in one area. But if you throw in a food source, as we see in B, you see it make a beeline with runs, and then little flips, and more runs until it reaches the attractant. It's a very neat procedure. Spirochetes work a little bit differently because their flagella are inside, so their movement is more like an inchworm. Some bacteria contain genes that allow for the production of bristle-like structures that enable them to adhere to surfaces, particularly tissues. E. coli is notorious for producing fimbriae and being able to attach to the urinary tract tissue and aids them in causing an infection. Pili are useful in conjugation, which is a means of transferring or exchanging genetic information. It's a means of sexual activity, if you will, between bacteria. During the process, a bacterium can shoot this pilus out like Spider-Man shoots his web and pull another bacterium closer so that they can give each other different genes. It's an interesting process that we will get into deeper later. Nanotubes can be used for communication or to transfer substances. In this picture, you can see both the fimbriae, which are darkly stained, and the pili. Some bacteria produce an S layer, which is simply tons of copies of a protein linked. A bacterium will produce this whenever they feel threatened. A glycocalyx is a sugar-rich structure that can be arranged a couple of different ways. If it's loosely arranged, it's called a slime layer, which helps the bacterium from losing cellular components. A capsule is more organized, and whenever a capsule-producing bacterium is grown on agar, it has a mucus, sticky-like appearance. These capsules help a bacterium to not only attach to tissues, but also helps them to get away from our immune system cells. Here you can see both the S layer and the glycocalyx. Figure A shows that mucoid appearance of a capsule-producing bacterium on agar, while B shows a capsular stain, the capsule being the halo or brightness that surrounds the bacterium. As I mentioned, a capsule is beneficial to a bacterium and contributes to its pathogenicity, which means it makes it easier for the bacterium to cause disease. It prevents our immune system cells from phagocytizing or eating it. Every time they try to grab it, it slides out like if you were to try to grab a fish with your bare hands. Biofilms are communities of bacteria and can be found on your teeth in the form of plaque. They're also the cause of many infections, as they can form on anything from catheters to pacemakers. Let's look at the biofilm formation. You can first see a few bacteria making their way to the surface of a catheter. Once they stick, they start to call all their friends to join the party, and then they reproduce and form an extensive barrier. This barrier makes it difficult for our immune system cells and antibiotics to penetrate. The cell envelope of a bacterium, depending on what bacterium we are dealing with, has 2 or 3 layers. Those layers are the cell wall, cell membrane, and outer membrane. Let's look at some differences in a gram-positive bacterium and gram-negative one. On your left, you will see a gram-positive bacterium, which contains a thick layer of peptidoglycan in its cell wall. You see the cell membrane, along with teichoic acid and lipoteichoic acid. In a gram-negative bacterium, you'll notice an extra layer, which is the outer membrane, which contains porin proteins and lipopolysaccharides. It has a thin layer of peptidoglycan and has a periplasmic space and a cell membrane. The whole purpose of the cell wall is to determine shape and provide a support system for the bacterium. A lot of antibiotics target the cell wall, which causes the cell to lose its support and rupture. The major component of a bacterial cell wall is peptidoglycan, which is held together by peptide bonds. A gram-positive bacterium has a significantly thicker cell wall than a gram-negative bacterium. Those teichoic and lipoteichoic acids help to maintain the cell wall and give it a charge. A gram-negative bacterium has a thin cell wall, which enables it to be a bit more flexible and more sensitive to rupturing. We talked about a gram stain, which is a differential stain. Let's look at why we see different colors in a gram-positive cell versus a gram-negative one. Our first step is the addition of crystal violet. After this step, both cell types would be purple. Our next step is to add iodine, which helps to affix the purple in the cell wall. The gram-positive cell will retain a deeper purple because it has that thick peptidoglycan. Both would still appear purple. Next is the most crucial step, which is the addition of alcohol, which dissolves lipids in the outer membrane and strips the color from the cell wall. After this, you can see the gram-positive cell retains the purple, retains the crystals and still appears purple, while the purple leaks out of the thin cell wall of the gram-negative cell, so it would be colorless. We add the safranin so that we can visualize the gram-negative cells. The gram-positive cells are unaffected, so stay purple, and the gram-negative cells pick up the color and appear red. Some bacteria don't have your typical cell wall. Mycobacterium and Nocardia have peptidoglycan, but in addition to that, they have a waxy outer layer called mycolic acid. This makes it difficult for normal staining procedures to work, so the acid-fast stain was developed. These organisms cause diseases like tuberculosis and leprosy, which can be problematic. Archaeal cell walls are a bit different. Some contain solely polysaccharides, some are made of proteins, some don't have peptidoglycan, and some have no cell wall whatsoever. There are also some bacteria that lack a cell wall. Mycoplasmas never had a cell wall, so they rely on sterols for the support and stability. M. pneumoniae is one of these and causes walking pneumonia. L forms are those that once had a cell wall, but for unknown reasons lost it. These guys are responsible for persistent infections and tend to be resistant to antibiotics, particularly those that target cell walls. Let's look at the outer membrane of gram-negative bacteria. The lipopolysaccharides are the bad part of this outer membrane. They release endotoxins, which cause fever and inflammation. The porin proteins also allows the bacterium to control entry and exit of substances. The cell membrane is a phospholipid bilayer used for chemical reactions. As we know, the bacteria don't have organelles. It's also selectively permeable as well, allowing the bacterium to control entry and exit of substances. The outer membrane makes gram-negative bacteria more resistant to antibiotics and makes it more difficult to kill with chemicals. However, alcohol can break down the lipids, which is why prior to a vaccine, an alcohol swab is used. In order for a drug to work against a gram-negative bacterium, it must pass this outer membrane. The bulk of the cytoplasm is made of water, and it is where a significant amount of reactions takes place. The DNA of bacteria can be found in chromosomes, which are stored in the nucleoid. There are also plasmids, which are not essential to the survival of a bacterium, but can be used for its benefit as an antibiotic resistance. Ribosomes are essential for protein synthesis and are made of ribosomal RNA and proteins. In a bacterium, it is a 70S ribosome compared to an 80S ribosome in a eukaryote. Inclusion bodies can be used for storage along with gas storage to allow a bacterium to be buoyant in its environment. Microcompartments are used in a variety of biochemical reactions. The cytoskeleton provides shape for a bacterium. They're structurally different than in a eukaryote, so may be a target for antibiotics in the future. Endospores are produced by members of Bacillus, Clostridia, and Sporosarcina. These guys are dormant, meaning they're not metabolically active like a vegetative cell. The endospores are produced when a bacterium feels threatened and can survive heat, freezing, and drugs. Let's look at the process. Number 1 shows us a vegetative cell who is starving. They then make copies of the genetic information and start to separate. We then see the cell develop a partition, which gives a forespore and a sporangium. Eventually, the sporangium engulfs the forespore. The forespore then develops endospore layers and then further develops into a mature endospore as the sporangium disintegrates. The endospore is then released, and during germination, a vegetative cell is released. Essentially, what happens is that the original cell realizes it's doomed without nutrients, so goes into hibernation mode to produce and release a new cell when the conditions are favorable. Some important endospore formers are Bacillus anthracis, Clostridium perfringens, which causes gas gangrene, and Clostridium botulinum, which causes botulism. We can't finish without discussing archaea. We don't hit it as much because as of now, no known members cause disease in humans. Even though they're prokaryotes, genetically, they're more similar to eukaryotes. We know archaea as being extremophiles, being able to survive in the most harsh environments. They are found on us and may have the potential to cause disease. This particular table is great as it helps to show you the comparisons between the 3 major domains. How do we classify these organisms? Berge's Manual of Systematic Bio looks at genetic sequences, whereas the Manual of Determinative Biology uses those characteristics that we can physically see. Taxonomically, there are 4 groups of bacteria and archaea. Gracilicutes are your gram-negative bacteria. Firmicutes are gram-positive, and tenericutes are those that don't have a cell wall. Mendosicutes are members of archaea. Anytime you see the term species, it refers to bacterial cells that share a significant portion of genetic information. Subspecies are members of the same species with some different traits. Serotypes are those that generate different responses from our immune system.